Alright, so for this video what I'm going to be doing is I'm going to be covering a couple of different bioinformatics tools, but really we're going to be focusing more on databases, and specifically the databases for Saccharomyces cerevisiae, or yeast, which can be found at yeastgenome.org. I'm going to walk you through a little bit of the functionality of the Human Genome Database tool, and finally I'm going to spend a good bit of time on the Arabidopsis thaliana repository, also known as TARE, the Arabidopsis information resource. So sit back and learn a little bit about these tools and see the access that you have whenever you're working with them. All right, well, thank you very much. So the first database that I want to show y'all is the Human Genome, homepage. This is housed at the University of California, Santa Cruz. And this is a tool that is helpful because you can go through and you can look at a genome and look at and search for a gene and find where it is located. This is the same data set that is housed at the NCBI. So in the event that you're ever looking at the NCBI webpage and you're looking at this exact same database. So I'm going to go to the Genome Browser, and what you're able to do is select different organisms. So this gives you you could start with the human genome, but you could also scroll through and, maybe go on to look at something like S. cerevisiae or Saccharomyces cerevisiae, or the yeast genome. I'm going to stick with human though, and notice whenever I'm clicking different organisms, I want you to pay attention to this find position and see what changes. So I'll click on, or human is currently selected. If I go to rat, well, that's going to change from human assembly to rat assembly. And so what that is doing is basically giving me the most up-to-date, or it gives me a number of different genomes to compare, and those genomes are basically the most recently published ones and the kind of consensus genome sequences. So I'll go back to human, and as you can see, the most recent human assembly is from December of 2013. This doesn't have the original drafts from the late 90s, early 2000s, but December of 2013, I'm going to go ahead and I'm going to search for ACE2, and I'm going to click go. And what this is going to give me is a couple of different options. And that ACE2 protein is the one that is implicated as being basically the receptor to enable the SARS-CoV-2 infections to take place. And that enzyme is the angiotensin 1 converting enzyme 2. And I'm going to go ahead and click on that. And whenever I click on that, this is going to show a huge amount of information, and maybe not all of it is something that I really know what to do with. But one of the things that it's going to show you is the chromosomal location. If you look at my cursor, I'm kind of hovering around and circling that. And then if you look around, so we've got this depiction of the chromosome that we're going to find this protein on, or sorry, this gene on, and it's going to give the specific location. It's going to give me chromosome X from base pair, that would be 15,561,33 to 15,602,069. And so that's found within a range that is approximately 41,000 base pairs. Now what I can do with this is I can scroll in a little bit, or I can zoom in a little bit, and ultimately get all the way down to the base sequences. So if you look at the top here, where I've brought my mouse, my cursor, this is going to show me any sort of different haplotypes. If I move my cursor down, I've got an alignment of a number of different vertebrates, and if you see the top one listed is the rhesus monkey, then the mouse, dog, elephant, chicken, X tropicalis, zebrafish, and lamprey. So when you look down at closer to the bottom of this window where I'm hovering, or this subsection of the screen that I'm hovering, zebrafish and lamprey, those are both fairly low in terms of consensus sequences, or common sequence of my gene, and I'm deriving that based on this black marking that shows me the identical residues. So I'm going to click up at the top. There's a zoom in function. I can go 1.5x, 3x, 10x, or down to the base pair. So I want to look at the base pairs. And how is that going to change my screen? What that's going to do is where I had my alignment, that's going to show me an alignment of all of the different organisms again. But it's going to be showing me the individual base pairs. And as we look at human versus rhesus, well, there's pretty much identical sequences between human and the rhesus. Now, what that means in terms of the protein sequence is a little bit, well, at this, what this is showing is not, well, it's not showing that information, but since we have a very homologous sequence between human and rhesus, that's why those rhesus macaques would be the target organism that we want to investigate for any sort of studies of SARS-CoV-2 infection. So the human genome browser does require a little bit of a, well, let me be clear, any sort of genome analysis that you're doing is going to require some background information. You're not going to necessarily just go and download a PDF or a Word doc with the human genome sequence. I suppose you could access that, but you know, if you think about the number of base pairs that are involved in that, it's just an overwhelming volume. But if you look at, if you have a target that you're looking for, a protein or a gene or something, you're going to be able to narrow your scope a little bit. So here's the human genome tool, and basically one of a very small amount of information that is currently, or that is available for you that you can quickly access. Now this next database that I'm looking at, and I just want to be clear that the last source that we were looking at, we could access a number of other genomes and kind of be doing blast searches of those. But the only reason that I'm kind of dividing this up is because I want to show you the different pages where these, or the different kind of landing pages that these different genomes are found on, because they do provide unique tools. So if you do research on yeast, on Saccharomyces cerevisiae or any other yeast organism, you're probably going to be using the Saccharomyces genome database as kind of your landing page, just because it's organized for this specific organism. And so what you can do with this is you can go ahead and you can do a fungal blast. You can look at different species and strains of Saccharomyces, and you could, heck, here we go, download the genome. And this is what this is going to show you is the most recent genome releases of Saccharomyces cerevisiae, or Saccharomyces. So in addition to housing this sequence data and releasing this sequence data on a pretty regular basis, one of the things that you can do with the SGD, as it's commonly known, the Saccharomyces genome database, is you can go to play with some of the tools that they have. For instance, I've gone over to function, and I'm hovering over that, and I see biochemical pathways and phenotypes. What I'm going to do is I'm going to browse all phenotypes. And I've opened a new tab for that. I'm also going to look at biochemical pathways. Now, with biochemical pathways, what I can do is I can look at some different molecules that are involved in different metabolic processes. And so what I'm going to do is I'm going to go to dead end metabolites. And what this gives me is a definition, first of all, so I know what I'm using. A dead end metabolite are metabolites that are either only consumed, or only produced by the reactions within a given cellular compartment, including transport reactions. Although some DEMs are biologically accurate, many DEMs aid in identifying incomplete or incorrect curation in the PGDB. Now, what I've got here is I've got some different options that I can use. And what I want to do is I want to just include all molecules as potential DEMs, include non-pathway reactions in my search, and treat reactions with unknown direction as bidirectional. So basically just go with the default. But what I want to do is I want to change my compartment from the cytosol to, let's go with the mitochondrial lumen. It says there's only one there, so I want to see what that is. Submit my query. Lo and behold, well, my dead-end metabolite, and if you're taking metabolism, or if you've taken metabolism, well, it's not too radical to see, and I'm just scrolling through to show you that there really are not too many different DEMs, but they are coenzyme A and L-carnitine. Now, if you've taken metabolism, you know that coenzyme A is involved in the transport of 2 of the 3 carbons from pyruvate into the mitochondria for subsequent oxidation, and carnitine is involved in the carnitine palmitoyl transferase or fatty acid breakdown. But this just, so anyway, this shows us 2 potential dead-end metabolites. But obviously we can go through and look at, maybe a choke point reaction. And a choke point reaction is a reaction that is either a unique consumer of a given metabolite, the only reaction consuming that metabolite, or is a unique producer of a metabolite. That metabolite must have at least one reaction that consumes it and at least one reaction that produces it. In other words, the metabolites must not be dead end. In theory, choke point reactions make good drug targets. So this is a tool that basically gives you a mechanism to look for a potential drug, because disabling that reaction disrupts the only path to or from a metabolite. Okay, so I'm not going to change anything. I'm including reactions found in humans, including reactions catalyzed by more than one enzyme. I'm going to include all reactions. Okay, so submit my query, and lo and behold, here's what I've got. I've got a reaction. I've got a couple different reactions, and then the corresponding choke point compound. And so then the third column is going to be the pathways in which a molecule is involved, or the pathway in which that molecule is involved. And so if I look at the fourth one here, this is involved in leucine biosynthesis, and it's a part of the super pathway of branched chain amino acid biosynthesis. My molecule is 2-isopropyl 3-oxosuccinate plus a proton yields 4-methyl-2-oxopentanoate and CO2. So if I click on that, it's going to show me my reaction that is actually taking place, and this reaction direction is shown in accordance in which the direction was curated. Most biosic compounds have been protonated to a reference pH value of 7.3, and this reaction can occur spontaneously, no enzyme is required, and it also gives me the Gibbs free energy for that given reaction. So that's if you're looking at metabolism, dead-end metabolites, and choke point reactions. Now, another kind of offshoot of this that we previously pulled up, and here's my other tab, which was my different phenotypes. Okay, so this is going to give me a map of basically different phenotypic categories and how you can kind of look at them. So what I want to do is I want to look over in this top portion, and actually, do I want to look there? Now I want to look in this bottom at about the 7 o'clock position. I see filamentous growth, development, budding, sexual cycle, and lifespan. I want to click on lifespan. Okay, so what this shows me is potential mutations, or the phenotype of a change in lifespan from the kind of average lifespan of a yeast molecule, or of a yeast cell. Now, if I click on the, or if I look at my ontology diagram, well, that lifespan can either be, sorry, if I look up at the top here, I've got qualifiers: abnormal lifespan, decreased, or increased lifespan. I'm going to click on increased lifespan because that's going to narrow my search from just something that changed the lifespan to an increase in the lifespan. Okay. Now, what this gives me is all of the different genes that have been implicated in causing a change in the lifespan of a yeast cell. Now, what this shows right here is I've got my gene, the phenotype, the experiment type, mutant information, strain background, the chemical that was used to treat this, and details of it. So if I look at my first one, oh, and the final column is the reference, so the paper that this information originated from. So if I look at my first column, I've got ABZ2. That's my that is my gene, and that's pretty common of yeast proteins or yeast genes. It's a series of letters plus a number. So we've got ABZ2, ADR2, ADR1, AIM46, and what we see with this is ABZ2. I'm looking now at the details of the experiment. A brief summary of this is that cells stored on YPD plates, that's the media that yeast is generally grown on, at 4 degrees for 5 years. Okay, so these cells, they survived for a total of 5 years, which from my own personal experience, that's a pretty dramatic spike or increase. Now, if you look at the final column, that gives me my reference, the paper that it originated from, and I can click on that, open a new tab, and this will ultimately give me the abstract for a paper and a link to the paper itself. So, surviving in the cold, yeast mutants with extended hibernating lifespan are oxidant sensitive. So this is the original paper that you can look at. This is from 2009 to see where these genes, or get more of the details on how these genes influence lifespan of yeast. Now, what I want to do is I want to go on to try to find some proteins that are from a different paper, because if you look in this, I'm clicking 1, 2, 3, 4 at the bottom of the screen here. Now I'm on the 5th page, 6th page, 7th page, 8th, and they're all basically from that exact same paper. So let's see, does this continue? I think I saw one on the 7th page. Yeah, 7th page. This TSA1 from Hansen S et al. 2016. I'm going to click on the paper just to see what I've got. So caloric restriction extends the lifespan of flies, worms, and yeast by counteracting age-related oxidation of H2O2, scavenging pararedoxins. Here we show that the increased dosage of the major cytosolic PRX in yeast, TSA1, expands lifespan in the HSP70 chaperone-dependent and CR caloric restriction independent manner without increasing H2O2. So this paper is all about the overexpression of this particular protein, or this particular gene, and how that resulted in an increased lifespan of these yeast cells. So that's pretty interesting, and this is all publicly available information. So then, you can go back. That was on page 7. Yeah, 7. I can click on this gene, and get a little bit more information about it. A brief description: a thioredoxin peroxidase acts as both a ribosome-associated and free cytoplasmic antioxidant. And as you scroll through, it gives me some information about the protein. It's 196 amino acids. The protein, when expressed, is 21,583.8 Daltons. Has an isoelectric point of 4.77. Then some of the connections that it may have through cellular components, so where in the cell is that protein found, biological processes, what is that protein involved in? It's involved in, for instance, DNA damage checkpoint, DNA protection, so all of the different things that this protein does. So the next database that I want you to be exposed to is the Arabidopsis Information Resource, or TARE. This is the resource that, or this is the host for the genome Arabidopsis, or for Arabidopsis, and specifically kind of the one that you're maybe most familiar with is Arabidopsis thaliana. This is the resource that I have the most familiarity with. And I think that it's a really cool one because there's tons of information that's available to it. And what I'd like to do is kind of introduce you to some things. So if you look at the top of the screen here, I'm going to zoom in quite a bit. You have a place that you can search for a specific gene. Now, Arabidopsis thaliana has a naming scheme that follows a particular pattern. So the way that you search for an Arabidopsis thaliana gene on TARE is by first typing in AT. Now that AT stands for Arabidopsis thaliana. The next thing that you're going to type in is the location on which chromosome an Arabidopsis gene is found. Now, I don't expect y'all to know all of this information off the top of your head or anything like that, but as you study genes in a specific organism, you become very familiar with different aspects of them. The gene that I studied as a graduate student had the, I guess, number naming system, location system that was AT2. So my gene was located on the second chromosome of Arabidopsis, and it had a genus location of 44830. So that's kind of the TARE identifier for the gene that I study. And so whenever I search that, it's going to bring up basically all the information that's currently available about this. Now, my locus was AT2G44830. This gives me a little bit of a description of it. I'm going to go ahead and proceed. Now, my description is, well, this is a gene that is a protein coding gene. My description is this is an AGC8 kinase involved in the pulse-induced first positive phototropism, plasma membrane-associated element of a molecular rheostat that modulates auxin flux through developing protofloem, sieve elements, PPSEs, while interacting with BRX, thereby timing PPSE differentiation. This also activates pin-mediated auxin efflux. Okay, now that's a lot of jargon. What I'd like y'all to know, and what I think is kind of the easiest pieces of information, is that this is codes for a protein kinase. Now, a kinase is something that you're familiar with, having taken Chem 4375 or 3375. It's a protein that will phosphorylate another protein with the help of ATP. Now, if you look below this, if you look below the description, you'll see other gene models. And it gives me AT2G44830.2 and 0.3 respectively. These are different splice variants. What that means is that going from your DNA sequence to the RNA sequence, there are slight variations in those different genes, or in the protein products that are ultimately produced. And so this shows me, oopsies, what I just disappeared, or what I just eliminated, was those different splice variants. Now, while that's loading, which will take a few seconds as it did just a moment ago, I wanted to show you that below that map detail image is different annotations. So based on the sequence of this gene, what is it potentially involved in? What can it possibly regulate? And some information about when it's expressed. So this is all like developmental information from when that protein is, or when that gene is actually going to be expressed and what that protein does. Now, bear with me for a moment, it continues to load. Oh, there it loaded and scrolled. Okay, there we go. So here are those three different splice variations, or splice variants of this protein. And what's really kind of, or it's not particularly important that you're familiar with that, but the tear resource, what I really like about it is this shows, so this bar EFP browser, if you look in the lower right hand corner, or lower left hand corner, I'm sorry, it shows you a kind of a heat map of where this gene is expressed in different cell types. So we've got a range from 0.0 to 30.35. And that 30.35 means that it's expressed in a higher concentration, or there's a more of it being expressed. And if you look at all of these different, so looking at the top left-hand corner, we have days after soaking, so seeds during germination. So this first picture is, or this first portion of it is dried seeds, Arabidopsis seeds, before you've done anything to them. So they have not begun any sort of germination. Then what you're going to do is you're going to get them, like put them in a damp paper towel or something, or water them. And what's going to happen to those cells, or those seeds, I'm sorry, is that there's going to be a germination process where the seed will actually begin to develop, and you'll see very early stem cells begin to develop from that. So after that, we have basically this entire kind of lifespan of these seeds. So 1, 2, 3, those 3 phases during germination. And then we have a 1-day-old seedling where we're looking a little bit closer. And the cool thing about this is data from an experiment that somebody actually did, where they separated in a 1-day-old seedling, which if you've ever seen an Arabidopsis seedling, it's about, I would say, 10 times the size of a grain of salt, so, or grain of sand or something. It's extremely small. Even being 10 times that size, it's still extremely small. But somebody separated the root from the remainder of that 1-day-old seedling. They did a DNA and RNA extraction, and they looked at the transcription of this gene and of every other gene. This gene ultimately is not expressed at a very high level, and all of this data is transcription data. It's not expressed at a very high level. If you look at the young flower or the mature flower, you have a little bit of expression that's a little bit higher than in other locations, but ultimately this is not expressed at a very high level. Now, if you scroll through the remainder of this page, but sorry, I don't want to lose focus, that this is just one example. All of this is going to be showing you where different genes are expressed and kind of what where they're expressed at different developmental stages. This also provides you with protein data. For instance, the protein product is 765 amino acids long. The molecular weight is 84,000 Daltons, and this protein has an isoelectric point of 6.73. So as you scroll through, this also has recent publications on this paper, or on this gene, and this shows that in 2020 there was a paper that cited this gene in particular that was involved, it was published in Nature Plants, and it's involved in auxin transport. Auxin is a plant hormone, and so if you click on that link, it will take you to, well, here's the information about this paper, and it will give you the abstract and everything like that. Now, so that's one kind of direction that you can take with this resource. Another direction that you can take is, I'm going to write Arabidopsis. I can go back to the original page and look at the other resources. This Java item seems, or this Flash item seems to be taking a long time to load. What you can also do is go and look at microarray data. So microarray data is transcriptional data. So when is a gene expressed and under what conditions? And so this is a TARE microarray experiment search. So when somebody did an experiment to test the transcription of basically every gene, they submitted their data to TARE. And so here what we can do is we can search by keywords, experiment goals, variables, plant tissue that was isolated, or we can look by experiment category. And I'm going to say that we look by experiment category, and let's look specifically at chemical treatments. When we submit our query, this is going to show us all of the different, as it turns out, there are 32 submissions where somebody did a chemical treatment and did analysis of the expression of the different genes in Arabidopsis. And so the first one is aluminum stress. Okay, so let's go ahead and click that. So soluble aluminum 3+, a specific aluminum cation, is toxic to plants. So Al stress is a major limiting factor for crop production on greater than 30% of the world's arable land. How plants respond to Al stress is not well understood at the molecular level. While our understanding is better at physiological and genetic levels, this is a proposal to screen Al-inducible and repressible genes. So this was a search of what are the genes that are induced in response to aluminum, and what are the genes that are basically repressed in response to aluminum. So this description goes on to tell a little bit about the experimental procedure. Then if you look at this tab, Slides and Data Sets, you can actually access the original data set. And so what we can do is we can say Get Data. Click Download. And whenever we download that, it's going to give us a what's called a RAR file. And within that RAR file, I don't know, I've always called them RAR files, we have 3 different text documents. Okay. Now, what this shows is a huge amount of information, and it's kind of complicated to process. But what it's going to do is it's going to give you specific genes. So AT2G42880 was analyzed in this study. And what I'm looking for whenever I'm looking for this, or looking through this, is did the expression of this go up or did it go down? And so what I'm going to do is I'm just going to copy that, and I'm going to look at that gene in, so sorry, the point is, whenever you have this, now you have a data set that you can look at you can look at what genes have been implicated as being expressed or, well, not expressed in the presence of aluminum. I'm going to close this. I'm going to go to a different data set for a different organism, or sorry, for a different treatment. This next one was Antomycin A treatment. I remember I looked at this data set. So this was detached Arabidopsis leaves were incubated with 10 millimolar Antomycin A in 1/10 strength MS media, and that's a sugar media for the cells to grow in, for 30 minutes and 4 hours in the dark. Control leaves are not incubated. Okay, so if I go to my first data set, what this is going to tell me is, just a second, got my original file, my text file there. Oh wait, no, I want to look at tear. Okay. Okay, so when I look at this tear file, this shows me my locus, AT2G36880, and it gives me all of these columns. This looks like it's a tab delimited file, so we could change the formatting by doing some tabbings, make it look a little bit better, a little bit more organized. I'm not going to spend too much time doing this, but what we've got then are questions of, is it expressed? Yes. And then a fold change. So comparison to the wild type or the control cells or the control tissue. How did the expression of this gene change? If we look at AT2G36880, I see a fold change of 1.105. Below that, I'm looking at locus AT3G50370. I see a fold change of negative 1.038. Now, when I look at those numbers, well, that tells me that some are upregulated and some are downregulated. Now, let me do this. I'm going to do the same thing, but with that original, where was it? That original file that I had for the, wait just a second, there we go, my aluminum stress. I'm going to go back to my aluminum stress treatment, slides and data sets. I opened the wrong file. I'm going to download this again, open this. I'm looking at my tear file, close. Okay, so I've got, just going to clean this up a little bit. Okay, so if I look at my, I guess that would be my fourth gene, AT2G. This is a gene that's on the second chromosome, 15240. This gene is expressed in my untreated, this is back to the aluminum, the aluminum treatment. It is expressed, and my fold change was 1.091. Okay, so whenever I go and I look at this gene, I can see that it is upregulated slightly. And this gives me another gene that I can begin to investigate. And the cool thing about this is this might be something that nobody has looked at all. You've got all this information that basically gives you a starting point for tons and tons of different research projects. That's one of the best things about these high throughput analyses, where someone's done genome sequencing and they've done all the transcriptome analysis. The cool thing about this is that, heck, look at this. Our most recent publication related to this gene was in 2008. So it's more than likely that no one has touched this gene or no one has investigated this gene in 12 plus years. and no one's really narrowed their focus specifically on this gene. So when you think about these high throughput systems, and it's really helpful to use them as great repositories of huge volumes of data. So those are the three databases that I wanted you to be familiar with. I wanted you all to be familiar with the human genome, the yeast genome, and the Arabidopsis genome. Now, in just a moment, I'm going to play around with some simple bioinformatic tools for DNA and RNA analysis.